Joining us this afternoon for our seminar focused on research and treatment development for SCA3 or MJD. This is part of our new webinar series in which we're spotlighting a single disease each month and providing webinars for both clinical care and the current state of research. So I hope many of you were able to join us earlier this week for our talk by Dr. Jennifer Faber on the clinical care and diagnosis of ataxia. And specifically, SCA3. So today we're joined by Dr. Hayley McLoughlin, who's going to talk about the research and treatment development for SCA3 and MJD. So before we get started, I'm just going to share a couple housekeeping items. If you can go to the next slide. Sure. All right, so if you require or would like closed captioning, that is an option, and you can do that by selecting the CC icon from your Zoom menu. You're feel free to chat with other attendees using the chat feature, but please, if you have any questions for our speaker, please use the Q&A icon at the bottom of the menu. We're only going to be answering questions that are coming from Q&A, so please pop it in here. We'll try to leave about 15 minutes for questions at the end of the talk. And just a reminder for those who did miss the webinar earlier this week, it's already posted on the NAF website at ataxia.org/webinars. So you can check it out there. All right, well, I am very excited today to introduce Dr. Hayley McLoughlin. She is an assistant professor in the University of Michigan Neurology Department with a joint appointment in the Department of Human Genetics. She did her PhD with Dr. Bev Davidson and her postdoc at Hank Paulson's lab, which coincidentally is where I did my PhD, and I had the privilege of working with her at the University of Michigan as we focused on SCA3 and SCA3 drug development. So she's now an assistant professor on the tenure track, and her lab is focused on understanding disease mechanisms, developing gene therapy approaches, and discovering disease biomarkers for hereditary ataxias, including SCA3. She has a passion for science outreach and is also a co-founder and managing editor for SCA3, a blog that is now become partnered with the National Ataxia Foundation. They're really interesting and really outstanding resources can be found on our website. So with that, I will go ahead and welcome Dr. Hayley McLoughlin.
Thank you very much, Lauren, for that kind introduction. I've just been monitoring the chat while I've been hearing the introduction, and there are those joining us from India, from Canada, from Brazil, from Germany, from Rome. I'm thrilled to be able to speak to you today about SCA3, all the research, all the therapeutic opportunities that will be coming down the pipeline, hopefully in the near future, what you can do as both a patient advocate or a family member of an SCA3 patient to learn more about this disease. So a little bit about what we are going to be covering, and just as a point, I'm not going to monitor the chat during this time. Lauren will do so at the end of this session. So if you'd like to hold your questions or add them to the chat at the end, I will gladly get to them. So I'll keep it within about 40 minutes or less to chat about some of the agenda items here. Okay, so what we're going to talk about today, an overview of the biology of SCA3 or MJD, Machado-Joseph Disease. We're going to talk about how researchers really study this disease, get into the nitty-gritty of some of the modeling, what I do, what's the bread and butter in my laboratory, and many other researchers across the country, across the world, who study this disease. We're going to talk about therapeutic approaches, both preclinical development and some of the potential biomarkers or opportunities that will help make these therapeutic approaches move forward to the clinic. And then I'll touch briefly on the current state of research and clinical trials, and also talk about the next steps if you've been diagnosed. And this really brings it back home to the NAF and how you can start reaching out with the resources that both SCA3 has created in collaboration with the National Ataxia Foundation, as well as the patient advocates that you've already heard from Lauren and from many others at NAF. Okay, so the biology of SCA3, and I'm going to be brief. I can by no means in 40 minutes or less cover everything on SCA3, but this is just to give you feelers here and give you at least a ground to stand on when you talk to your clinician, your neurologist, when you talk to those at NAF, or when you talk to researchers like me at National Ataxia research conferences in the near future. Okay, so the biology of SCA3. As you well know, SCA3 may stand for Spinocerebellar Ataxia type 3. You might also know it as Machado-Joseph Disease. This is a progressive cerebellar ataxia, and I just pulled these common features from ataxia off the National Ataxia Foundation website, which works pretty well for most of the ataxias, including SCA3. Now, SCA3 has speech disturbances and eye movement abnormalities, often blurred vision and double vision, gait clumsiness, gait problems, and progressive ataxia. There's less common for tremors, but there is issues with eating and swallowing and overall lack of coordination in general. Now, you might also know that SCA3 or MJD is the most common among these dominantly inherited ataxias. And I just pulled this recent review in Neurotherapeutics, highlighting in this, I put an asterisk on it because the colors are a little challenging, across the world how common SCA3 is. It accounts for basically 30 to 40% of the currently diagnosed SCA patient population, with about 140,000 or less than 10,000 patients in the US. Now, SCA3 is caused by a CAG repeat expansion in the ataxin-3 gene, and you've probably heard it referred to as a polyglutamine disorder. And so we'll talk a little bit about that. Now, what does a CAG repeat expansion mean? Across all of our genomes, there are repeat expansions between the nucleotide or DNA content. And it just so happens in ataxin-3, there's a repetitive sequence with a C, an A, and a G repeats multiple times. Now, the average person will have 2 copies of ataxin-3 with the repeat expansion between 12 to 40 repeats. One of my alleles is 17, if you're interested. But when you get this to the RNA level, this turns into a CAG in the disease transcript. And if you have this disease, it hyperexpands with above 54 repeats upwards to 87 is the highest reported to my knowledge. And it's one of your 2 ataxin-3. And the bigger, the earlier the onset of ataxia. Age of onset.
Sorry about that. I think she must have cut out for just a minute. So we'll give her 1 more minute to jump back on.
Yes, I'm back. Shall we try this again? Let's give it 1 more shot. If we have any challenges, and I apologize for the technical difficulties. For some reason, my internet at the University of Michigan has been spotty lately. Okay, so I think we got to, hopefully, and Lauren, chime in if this isn't where we were at, but the repeat expansion is variable. And the larger the repeat size, the earlier the age of onset in these patients with SCA3. Now, when you have a repeat expansion, this leads to a polyglutamine repeat in the protein ataxin-3. And when you have many of these proteins come together, they will aggregate. They will cause challenges in the garbage dumps of the cells. They will cause alterations to the DNA code and cause it to have challenges in repairing the DNA, which is unhealthy for a cell. There's going to be some circuitry challenges, and then there's going to be some energy challenges. All of these are well established across model systems, and I'll talk to you about those in just a few minutes. But all of this accounts because of this toxic gain of function of this new protein that has this hyperexpanded repeat size. Now, ataxin-3 normally is expressed throughout our bodies, throughout all cell types in particular, and throughout the brain. And so if you took the brain and set a cross-section through it, the pink here is highlighting in all the brain regions where ataxin-3 would be normally expressed. Now, a normal person would have this expression pattern of ataxin-3 throughout. If you have a hyperexpansion repeat, where it gets to this high CAG content in the ataxin-3 gene, you have actually regional vulnerability. Now, ataxin-3 is still expressed throughout this brain, but these brain regions, which are known as the brainstem or the cerebellum of a human brain, have vulnerability or challenges. And so these are the brain regions that you'll see phenotypes like aggregation or problems with the garbage dumps of the cells or DNA repair challenges. And one of the hallmarks of this disease is actually this nuclear accumulation. And so you'll see this throughout the model systems as something that we can interrogate for some of the cellular stresses that might be seen as ways to modify the disease. So how do researchers study this disease? And I'm going to tell you just a smattering of all the different types of techniques that my lab does, that other labs that I've worked with or collaborated with or have heard of from in the field. And where we start is with the patients, because that's our foremost interest here. So what we're looking for is a recapitulation of what happens in a patient population with SCA3. We can make mouse models that have the same repeat expansion in this mouse genome of ataxin-3. We can overexpress human ataxin-3 in the mouse. We can knock out ataxin-3 in a mouse. We can also overexpress versions of ataxin-3 in different model systems like a fly, a worm, and a fish. We can interrogate this through non-invasive imaging or through functional behavioral battery. And so in this case, we can look at it through MRI or MRS, and I'll tell you a little bit about what we do with MRS. We can look at a mouse in a chamber and watch them move around for 30 minutes and see how much they move on a total motor score. We can interrogate tissue samples. So I'll talk to you about some histology from postmortem tissue in patients and what we've learned from those data. Biofluids, such as plasma from your blood, CSF from your spinal fluid. Brain regions of mice that are, again, modified to mimic the disease state, or even cell culture systems where we take human skin cells or blood cells and we reprogram them into cells of interest, into stem cells that we can make into neurons. And I'll touch really briefly on that as well. And finally, all of these samples here, we can interrogate in many biochemical ways. We can ask how these proteins are being changed and what things are they interacting with? How are they challenging the other genes that need to make proteins in our cells? How does that readout translate to firing frequency? How does that readout look like on a cell in a cell dish, and which cells are they actually targeting? And so there's lots of ways that we can look into this. And I'm just going to start with kind of the basics. Back in 1998, in a Cell paper by Nancy Bonini, they created a fly model which overexpresses this polyglutamine ataxin-3 gene. And again, in a fly eye, it's actually a really cool model system where you can interrogate whether there's toxicity or unhealthy cells, basically, in the event of a protein that's toxic, like ataxin-3. And so comparing that to, again, ataxin-3, but with a smaller repeat, so something like a normal gene. The same can be tested in a C. elegans or a worm model. And you can see this aggregation phenotype that would be occurring when you have a repeat that's quite large in a worm. So between 75 polyglutamines or 130 polyglutamines. More recently, out in Australia, they've created a zebrafish model, which is a fish that they can interrogate, which is really a beautiful model because of imaging modalities. It's actually almost translucent at the larva stage, so the early states. And you can also test from behavioral assays in all three of these cases. And here's just how much less a zebrafish swims because of the repeat expansion in this zebrafish model. So these are very exciting, interesting invertebrate or vertebrate fish models that you can look at some of the basic biology that also translates to behavioral phenotypes in these model systems. More commonly, and what I use in my laboratory, is mouse models. And so my favorite to work with is this full-length human ataxin-3 diseased gene overexpressed into a mouse. And so it has what you and I have in our genome lopped in randomly into a mouse genome. And it has, in this case, an expansion of at least 84 repeats, which is within the disease range. And excitingly, this mouse model, when it was first generated back in 2002, has phenotypic characteristics similar to a patient. And so you would see in brain regions that are regionally vulnerable, in disease, nuclear accumulation of ataxin-3. And in this case, this is a brain region that you can image by staining for it with increasing levels of this mutant protein in brain regions that are similar, that will eventually, the cells will die in a human brain that has SCA3, hyperexpansion of ataxin-3. Also, these mouse models we can test on behavioral assays, like as they walk across a beam that's no more than 100 centimeters. How long does it take them to cross that beam? And we can test them over their time, over age. And so from 7 weeks to 55, 56 weeks of age, compared to a normal mouse that doesn't have this hyperexpansion of the ataxin-3 gene. And excitingly also, unfortunately, is they have a reduced lifespan. And so we can test for augmentation of this lifespan in that we can recover we can expand the lifespan or decrease mortality in these mice with a treatment. These mice also have the opportunity for us to do slice culture and actually directly patch clamp or interrogate through electrophysiology the signaling of cells that are demised in this disease. And so, for instance, in the cerebellum of a mouse, you have Purkinje cells. These Purkinje cells have a bunch of channels, and the channels within them regulate different ion conductance that allow for these electrical signals. And so in an SCA3 mouse, the firing frequency of a Purkinje cell fires a lot faster. And you can then sprinkle on some drugs and hopefully modify the expression or the firing frequency of those cells. Now, to put it a little closer to patients, let's start with the patient population. One of my colleagues, Gulin Oz from the University of Minnesota, is very interested in MRS, or magnetic resonance spectroscopy. And so you can look at neurometabolites within a brain across different brain regions of patient populations. And in this case, there's a bunch of SCA patients, including 1, 2, 3, and 6, relative to a control population. And you can interrogate over time in a non-invasive way how these metabolites change. And so levels of something like total NAA or total choline are related to different cell types within the brain that are challenged. And if you look at specific regions like the cerebellum that is demised or vulnerable in SCA3 patients, or the pontine region, which degenerates over time, we can get really interesting biomarkers or non-invasive opportunity to look at how these things are, how they could be measures prior to the onset of behavioral phenotypes in a patient or ataxia phenotypes in a patient. And excitingly, we can also do this in a mouse, with a mouse. And so using an MRI machine in the same way as you would normally for a patient, just a little bit smaller, you can actually image and look for the neurometabolite signatures in a cerebellum and a brainstem of a mouse that overexpresses the human ataxin-3 disease gene. And just like in a patient with SCA3, this is data actually from my lab that's currently under review. In the cerebellum of these mice, we have a differential expression that changes over time as these mice age of the total NAA, which is a marker of neuronal health within the cerebellum of these mice. I would be remiss not to talk to you about postmortem tissue. And there's been a tour de force by Rube et al. Throughout the last few decades, interrogating how different brain regions have neurodegeneration and looking at which cell types undergo severe neurodegeneration. And so in the case over here is just a schematic of this brainstem region. If you did cross sections, which brain regions are degenerating over time in patient populations. And this is really important because it gives us information about where to target, where do we need to deliver therapies once we figure out mechanisms of disease. Which brain regions are places that we don't want to target or don't need to get to, which is actually really important because getting into the brain a drug is one of the hardest jobs we have as neuroscientists, honestly. And again, I highlight just in this cartoon, the regional vulnerability of ataxin-3 is unlike the ataxin-3 expression throughout the whole brain And finally, I just want to touch on briefly some of the other stem cell modeling. And this is something that over the last decade is really coming to fruition, and it isn't yet a big piece of the data sets, as you might have noticed by the dates that have shown publications here. But it is a useful tool, both from understanding cellular phenotypes, understanding new ideas that could cause disease onset or pathogenesis, as well as opportunities to target with therapeutic intervention or gene editing, different cell types that might be affected within the brain. And in this case, I'm just showing a schematic of 3 different neural populations and how they can be reprogrammed from something as simple as your skin cell. Now, there's literature out there in many of these SCAs, particularly in mature neurons. There is yet to be a study on Purkinje cells and SCA3 neuron motor neurons. I would suspect that is coming down the pipeline. There is one FDA approved on the NIH registry, human embryonic stem cell, which can be reprogrammed into neuroprogenitor cells and programmed into cortical neurons, much like the skin cells. And those you can look at in the same context as you can from the iPSCs and show simple things like spontaneous firing, like what we can see in a mouse brain, but in the human context. All of these different opportunities allow for us as scientists different ways in which we can interrogate pathogenic mechanisms, therapeutic opportunities, which is forefront in most of our minds. So that leads me to therapeutic opportunities. What is happening in this field? What we know from all of these model systems, from postmortem tissue, from stem cells, from flies all the way up to mice, is that ataxin-3 likes to aggregate. Ataxin-3 has gain of toxic properties, and those toxic properties mess up a lot of the cellular functions, including transcription, some of the garbage dumps like autophagy, the powerhouses of the cells, the mitochondria, a lot of the signaling, like I talked about for electrophysiology. And nice reviews on opportunities on how do we actually counteract this is just silencing ataxin-3 or preventing that hallmark of disease, that nuclear accumulation or aggregation of ataxin-3. Some studies have looked at cleaving ataxin-3 and getting rid of this polyglutamine, which actually is at the back end of the gene or the protein. Others have looked at destabilizing it or helping to push it into these garbage dumps of the cells like autophagy or the proteasome pathways. And then moreover, there's been experiments or therapeutics that have just targeted the symptoms, like calcium signaling is dysregulated. So let's create an opportunity to try and normalize that. How do we, if there's glutamate hypertoxicity, how do we get glutamate back on schedule in the cells? And so these are all opportunities that are being explored across this world, across this country, whether that's through pharmaceutical agents, biologics, whether that's through gene editing or gene therapy. And so there's a lot of opportunity, and I'm going to talk to you a little bit about what's happening right now. And so I actually just pulled this off of the National Ataxia Foundation website. This is their SCA3 pipeline. These are the drugs that are currently in development. And I want to refer to this top panel here. So there are drugs that are not listed here in the discovery platform, and there are many. If you had attended the National Ataxia Foundation annual meeting this last November, you might have heard of many of these. There's lots of preclinical testing that's going on in these laboratories. How do we test these in all those model systems that I just briefly touched on? Finally, when you've proved that the cell can be modified by a therapy, that you prove that you can modify some sort of phenotype like behavior, and you've shown that this drug of interest is not toxic or off-targeting in a way that could eventually be damaging to the cell or to the organism or model, FDA gets involved, and you can file for a new investigational new drug. And that allows for you to enroll and start into a phase 1 clinical trial. And so you'll note there are a handful of drugs that are currently in clinical trials. I would say that Biohaven has really been the forefront for our field in establishing both the cohorts, the natural history development, and what we need to get organized for a clinical trial in this SCA field, as well as some of the other programs that are coming onto the forefront. And so I'm not going to speak to you about Biohaven because I think you might have already heard a little bit about that, and there's also press out there. And I'm not going to speak towards the stem cell-based therapies because none of those are going on in the US. And I think that there's press also out about stem cell therapies. But I am going to talk to you about the pipelines and what I'm most familiar with. And this is targeting ataxin-3 directly. So the upstream marker of ataxin-3, getting rid of it at its start. So before the aggregation or reducing the aggregation and downstream processes that can go wrong. There are many different drugs, and there's this really beautiful review that came out in Neurotherapeutics just a few years back that highlights some of these preclinical development drugs that we're looking into for SCA3. But it's really, what is the first candidate? What's the drug of choice? And then all of the work. So this is short-term safety profiling, and this is in different model systems. This is in cells, this is in mice, this is in iPSC lines, this is in rats, this is in non-human primates before it even gets into patients. Are there any chronic long-term side effects? Are there biomarkers? And that's one key topic that I'm going to highlight on and in the next couple slides. And then how do we show that it's actually targeting what we think our drug is targeting? I'm not going to talk about the clinical trial stuff because I think there's also a really beautiful overview of what the pipeline is for clinical trials, but I just want to tell you about how we get to a drug from the top of this pipeline and into the clinical trials, because US patients want to see as many things get to the clinical trial space and further. And so how does that pipeline occur? So that when you hear about it at these National Ataxia Foundation conference meetings, you understand this pipeline. Okay, so what you need to know, and I'm just going to speak about what my lab in collaboration with Dr. Paulson's lab and Ionis Pharmaceuticals and now with Biogen, how we've developed a gene therapy approach against ataxin-3. And so really it starts in science with the premise. And so in SCA3, in model systems, you can get rid of ataxin-3 from a mouse, and the mouse lives a normal life. They don't have any motor impairments. And you can also make a mouse and genetically be able to turn it off at about 5 months of age when they've already developed phenotypes of disease, and this reverses the motor impairment. So what this told us between these two model systems was that if we could turn off this mutant gene from being made, we could probably have a pretty powerful preventative therapy at hand. And so there's a couple ways to do this. And I was fortunate enough to be right time, right place when I started my early career training as a postdoc and now as a faculty to be able to collaborate on antisense oligonucleotides. And this is one of those FDA approvals for other diseases like spinal muscular atrophy. And I won't get into the nitty gritty of how it works, but suffice it to say, I'm going to talk about a gapmer ASO, and what it basically does is cuts ataxin-3 up, chops it into little pieces so it can't make a protein, this mutant protein that has all of those downstream events. There's other versions that get rid of the polyglutamine repeat, but I'm only going to speak about the gapmer design. And this was 2 papers that, again, I highlight the dates, was years ago. So we are now entering into phase 1 clinical trial with SCA3 on ASOs, but the pipeline is long and arduous, and that's to the benefit of making sure that we're doing the right thing, the right place for the right people. Okay, so what's this pipeline? First, you want to prove that you can have a target of interest, and then you want to target it in a model system in vivo. And is it active? And so one thing that we've done in our lab is we've taken a drug that is directly designed to ataxin-3 to chop it up, make sure it gets spread throughout the brain. And so this is a picture of this drug after we've harvested the tissue 4 weeks later and show that it's spread all throughout the brain. We've also then looked and said how much ataxin-3, this mutant protein, is left in those brain regions. And if you chop up a mouse brain and look at it from a biochemical perspective, we can knock down or get rid of over 50% of this mutant protein in a mouse brain. So that was the first line of evidence. Now you have to show that you can have a disease benefit. So what's the behavioral outcomes? Here's one paradigm we do in our laboratory. And this sounds pretty silly, but they stand on this clear platform, a mouse. They don't like to be in an open space. They want to run across this beam and go into this dark, empty, quiet room. And so how long does it take a mouse to go across this beam? And so if you treat a mouse, or prior to a treatment, you can run this assessment. It takes them about 7 seconds to cross this beam, give or take, in a wild type mouse versus a diseased mouse. Now the blue bar is the mice that are going to receive a treatment at 8 weeks. And then we're going to check them multiple times over the course of their life. We did actually repeat dose these mice with an ASO. And what I hope you can appreciate is that mice that should have developed like this red bar with an ASO therapy have actually reduced their levels of their motor impairments. This was huge. That meant that we had disease benefit. We went on with collaborators, with industry partners to push this forward. And this is the same pipeline that would happen for any other FDA-approved drug into larger model systems, rats for off-targeting toxicity, non-human primates. There's been so much work that I can't even speak about because I just don't have time or even, honestly, the wherewithal to get it all out. But what's important is that there has been due diligence on preparation for this drug and every other drug that has gotten past IND approval and into phase 1 clinical trials. And so SCA3 ASOs have now started enrolling for phase 1 clinical trial. But really where we're at, and this is what I think is going to be an important aspect of SCA3 and therapeutics at large and how they are beneficial to this community in the long stream, is the biomarkers. So I have to tell you, what is a biomarker? A biomarker is anything that has the opportunity to inform the disease process, how a therapy is fixing or causing more damage. And one of these is NFL, neurofilament light. And so this is a direct biomarker for neuronal health. It's actually emerging as one of the most promising biomarkers in neurodegenerative diseases. It's one of the most common tested across the clinical trial platforms. And in SCA3 patients, actually pre-ataxic, prior to the onset of motor challenges in patients, they actually have elevated levels of this neurofilament light protein in your blood. And so we can test by a simple blood test when you go from early to late onset, not based on your motor, or when you go from pre-ataxic to ataxic from your motor, but we can actually start to interrogate when an age of onset would occur for motor impairments. And this NFL levels are also directly related to your ataxia rating score. They are directly related to your CAG repeat. So the earlier the onset, the larger the repeat. I already showed you that. But the earlier the onset, the larger the repeat, the more NFL you have. So it's a really nice biomarker as an opportunity for future clinical trial platforms. And so again, this was just one piece of evidence on how to work your way through this paradigm or this pipeline. ASOs for the preclinical events to get us to IND enabling with some biomarkers and now into a phase 1 clinical trial. But I want to tell you about the current state of research and clinical trials. What are some of these other ones that are occurring right now? And there are many. Some of them are looking at glutamate regulation. And so that was from the Biohaven study. And glutamate is one of these proteins that is important for the electrophysiological signaling that we have in those cerebellar cells. We have stem cell-based therapies, and this is supposed to be a replacement therapy, an opportunity for the cells that are lost to have novel expression in cells that are in regions that are missing them, or for cells to provide factors that might have been secreted in the system as a replacement. Ataxin-3 reduction, we just spoke about that, and that is the Biogen study that's currently enrolling. There are a handful of studies that are also out there trying to keep ataxin-3 from aggregating. And then there's also work that's trying to keep ataxin-3 from having DNA damage and creating this expansion from getting bigger and bigger. And so towards those therapeutic opportunities, we need to touch on biomarkers just for 5 minutes or so. So what many of you, if you've been to see a neurologist, have done is undergone a scale for assessment and rating of ataxia. And this is your SCA3. They go through a bunch of motor coordination tests and they give you a score, 1 to 40. 0 meaning you have no ataxia, 40 meaning you have severe ataxia. Now, SCA3 is a beautiful opportunity for us to create a standardized platform across the world on how to interrogate ataxia. This is across platforms like SCA1, 2, 3, and 6. And over 1 year, SCA3 patients actually increase on average your SCA score of 1.4. And so over a 1 year course in a clinical trial, you have a 1.4 change in a patient expected. To put that into numbers on how many patients you would need to enroll into a phase 1 clinical trial, into a phase 3 clinical trial for efficacy, we need about 115 people that have early onset reversibility, which is a pretty big ask for rare disorders like SCA3. So this is my call to action. We need more biomarkers. In order to make these clinical trials move faster, in order to make the efficacy more reliable, and to give you guys the best outcome measures and the right therapy for your future treatment opportunities. And so things that are coming down the pipeline, if you go to clinicaltrials.gov, there's a study that's enrolling for wearable sensors. This is basically your Garmin's, your Apple Watches that they secure to your ankles and your arms, and they test how much you move around over the course of a day, over the course of a median time. And so just one outcome measure, and I apologize, I lost the citation here, that was just published last year, actually supports this. In the SCA3 patients, they just tested 23 people that have SCA3, that just looking at the angle of your toes, or how often you need both feet to stabilize yourself, can distinguish SCA3 patients from a normal control. And so this is a really sensitive measure. This is data points that SCA is never going to provide for us, that ataxia rating scale. And this might help normalize or shrink the error bars on how many patients we need to enroll in a study. Now, these are functional assays. These are biomarkers like SCA or wearable sensors that require you to have ataxia symptoms. But what about biomarkers before the onset of disease that correlate with disease progression? I already told you about NFL. But wouldn't it be great to have a biomarker that says you're about 5 years away from having ataxia symptoms? Now is the time to take a drug. Now is the time to fix those cells before they die off and we can't replace them. Anecdotal evidence is that Parkinson patients actually have something on the order of less than 75% or more of their dopaminergic neurons are already lost before they have their first symptom. And so there's not a lot of cells left to rescue. We don't know if that's the case for SCA3. We need to figure that out. And I think that those kinds of pieces of information are essential. And so if we can understand before the onset of behavioral ataxia symptoms, we might have opportunities on our trials to make them more effective. Okay, and then also we need biomarkers from biofluids that can show us target engagement. So is the drug doing what we think it's doing? And so in the case of like an ASO, we know it's supposed to directly target ataxin 3. Is it doing that? Are we reducing ataxin 3 in the CSF, in the spinal fluid, in the brain? That would be a remarkable biomarker. So there are many different types of biomarkers out there. Many of you have already undergone a diagnostic biomarker. This says, do you have the disease gene? Do you have this hyperexpansion? There are monitoring biomarkers. There are response biomarkers and predictive biomarkers. And these are ones that are going to tell us if you are going to be favorable or not with a drug of choice as we get more and more into this pipeline. Prognostic markers. When will you have the onset of your disease? Susceptibility, will this work for you? And then just safety biomarkers. Do we have a spike in NFL? if we have a spike in NFL from a therapeutic, that often means there's more neurodegeneration occurring, which is not good. That means there might be toxicity from the drug of choice. So there's lots of really informative opportunities from something as simple as a blood sample from you all, from a urine sample, from undeniably a spinal fluid sample. Those that are surrounding the brain, bathing the brain and providing us all this insight to the progression of this disease. There is lots of opportunities to join, whether you are US stateside or whether you are worldwide. So I encourage you to reach out to NAF or your neurologist and ask if you qualify. And this is just sites across the continental US and Ataxia Global Initiative, which is collating across the world, all of this data sets. And I can't encourage you enough to engage, because I think without patient engagement, we are not able to move this data, all the data that we're learning from our model systems forward. Okay, so with that, I just want to say thank you, and I really look forward to answering as many questions as I can in the next 17 minutes or so.
Well, thank you so much, Dr. McLoughlin, for that wonderful presentation. We have quite a few questions, so we're just going to see how many we can get through in the next 15 or so minutes. So one of the questions that has come up from a number of our participants is whether you think that a drug given at mid or late stage in disease might still have some efficacy in getting motor back and balance back if you're already symptomatic.
I think that's a great question. So this is the biggest challenge we have, and it remains to be tested, basically. For instance, and I can tell you from a scientific perspective, SCA3 mice, we treated them after the onset of behavioral symptoms. And so that's the gold standard, is we want this to be a treatment opportunity that isn't so far downstream that we have nothing to rescue, in that the cells are no longer present, but has opportunity to modify some sort of a behavioral phenotypic outcome in patients. And so I will say that most of the clinical trials are looking for early onset, just so that we get the best outcome measures to prove this forward. But from a evidence-based in science, how this works is they look for the easiest low-hanging fruit, and then they work broader across the patient populations. And so I think the opportunity is there. It's just a matter of the mechanism of the therapy. And so things like glutamate targeting or trying to activate the cells might be very beneficial, or just clearing up some of the aggregation or ataxin-3 accumulation in cells might be sufficient to make those cells work more functionally and alleviate behavior, even if it is late stage in disease.
Another question about ASOs for SCA3. Since ataxin-3 is expressed everywhere and it has specific functions in the brain, is it dangerous at all to be reducing the levels of that protein?
Can you say the question one more time? I got caught up reading one of the questions here too.
Oh,
I should stop.
Is there any danger in reducing ataxin-3 protein since it's expressed everywhere in the body and it does have specific cell functions?
Yeah, so that also remains to be tested. And so the one thing about ASO technology is that it's transient. It has to be repeat dosed. I think this is a really important aspect of that therapy. And it means that it can be dosed in at high doses or low doses. So it's not a one and done, just like you would have to dial in any sort of drug of choice when you go see your doctor. It's one of those, you might have some bad side effects and you just back down off the dose because you're having a repeat treatment. Now, towards this loss of function, that remains to be stated. Now, this is where human embryonic stem cell studies, human iPSCs and neurons, knockouts are going to be huge. We're getting there in the scientific community, but it hasn't been tested. Now, mouse models you can get rid of ataxin-3. They are arguably normal. They live the normal lifespan. They have no ataxia phenotypes. And frankly, from what I see comparatively in our overexpression, our gain of toxic function mice, it's a great life comparatively. So we can solve as much as we can, but it's a great first line of evidence from a targetability standpoint.
Thank you for that. And one more question about ASOs. Are those currently only available through clinical trial participation?
Yes, unfortunately. So this is the important aspect of research, is that it is going to be an ASO technology for other diseases, has arguably been challenged and also been really effective. And so if you follow the Huntington's field, they have canceled their ASO trial because of toxicity. Now, I guess backwards to the last question, Huntington's disease has a loss of function issue. And so that could be causing some sort of toxicity if you were to remove it directly, unlike ataxin-3. Now, is this therapy going to be available to everyone right off the bat? No. So it's going to be a dose escalation study. And I believe Biogen, NAF has a webinar, or at least a talk by the clinical trial phase 1 on how they are going about this enrollment.
And I'll just note that there is another ASO trial that'll be initiating soon in Europe from Vico Therapeutics. Much of what Hayley's presented today is from Biogen, who has the B132 trial. So there are a couple of questions about model systems. So one of them, there's obviously variability in patients and how they present and when they present and what symptoms they have. Do you also see variability in animal models of ataxia?
Yes, that's an excellent question, and it's something that we have to pay attention to. So we have the burden of a repeat. Even something as simple as a repeat size that modifies with the as mice breed, and they provide, just like when you procreate and have another child, and you pass on your repeat expansion or the size of my repeats, those are going to be modified, and that's going to cause different phenotypes. And so, for instance, in this YAC transgenic overexpression model, which is the one that we've done all these ASO trials within, the repeat size, when it gets too small, 75 repeats, we don't see as much of a behavioral phenotype. If it gets above 75 repeats, then we get an even stronger phenotype. So we've been very careful as scientists to really enroll as tightly regulated model system as we can, with an average repeat size of 75 repeats. We have model systems that are knock-in mice, and that we've gone and made this repeat really big, and it causes the disease to occur much earlier. Or really small, and we don't see phenotypes until over a year or two in mice. And so that kind of variability is definitely forefront in a lot of our experimental study and designs.
Just one more question on a model system. What does SCA3 look like in a fly? Do they have any similar symptoms that people do?
Yeah, they are ataxic. They don't move as well, basically. There's this really cool test where you tap it in, they're in a vial, and if you tap them, they just don't like to fly up to the top, basically, or crawl up to the top. So you can interrogate just the motor impairments in a fly. I will say the strength of the fly model is how quickly you can modify the genome. And so you can interrogate domains of this proteins, the little pieces of the protein, and how it regulates and interacts with other proteins. And so that is a real utility of the fly model. Additionally is the visual opportunity, which is that picture of the eyes that I showed you for toxicity. And so modifying the protein in its different interactions and saying which things are essential for this protein to interact with to cause this toxicity or not. What can we cover up to absolve this phenotype is really pretty cool.
Thank you for that. So a couple more questions about drugs. Do you have any comments that you want to make about some of the other clinical trials that are going on, whether that's the one initiated by Selos using the trehalose? Even if
I am
aware of the
less
mechanism of action.
qualified, honestly, to speak towards those. So I would feel more comfortable if a clinician who is actually a part of those studies spoke about them.
I appreciate that. And there was a couple of questions about CRISPR. How many years down the line do you think we are from an actual CRISPR-based therapeutic?
In patients? Oh, I think there is a lot of safety there. We just heard an excellent talk at the National Ataxia Foundation about gene editing and how we can modify and actually turn it off and be careful about when editing occurs. There's so much work to be had on how to get the right target, how to only cut that couple of times, how to cut the right cells, which cells do you want to target? And so again, towards that point that I kept trying to harp back on, ataxin 3 is everywhere. And we have to be careful about targeting cells that are currently resilient, that are they're doing their job and they're keeping everything going. So, and only targeting the regions that are actually demised and leading to these behavioral phenotypes that we'd all like to get rid of, these ataxia phenotypes. So I wish I could put a number on it, but I've seen this field grow. I'm hopeful. I think there's a lot of work being done. Right now, what CRISPR is doing for science is making everything move so much faster because we can do these genetic edits. We can change things from the genome level, and we can create model systems within a matter of months when they used to take years.
And I'll just note, Dr. McLaughlin's talked a lot about the ASO for SCA3. There already are FDA-approved ASOs for other neurological diseases, but there aren't FDA-approved CRISPR therapies for other neurological diseases. So
absolutely,
this isn't just for SCA3, it's for other diseases as well.
Yeah.
One question was about clinical trial design. Why do we have to have patients in clinical trials that take placebo instead of the real drug?
Yeah, I mean, it's an ethical dilemma sometimes, but without proper controls, you don't know the changes that are being had. And so there are those pioneers in every field that take the risk of getting a placebo. I will say through these clinical trials, it's placebo with if you are enrolled and this is efficacious, they will disclose and they will allow you to enroll for open label after for most of the trials out there. And so it's a catch-22 in the sense of whether you get the lucky hand of the draw, but you are essential to these studies. Without controls, science does not go anywhere. I start my graduate students, my technicians, my undergrads on controls before the science. Without a proper garbage in, garbage out dump of science, we can't really understand what's happening.
Thank you. We have a couple questions about the inheritance pattern and how repeat length affects SCA3. So the first question is, all SCA3 inherited?
Yes. So you will have a genetic diagnosis of SCA3 based on your repeat size. And I wouldn't bore you with you clicking back to the first couple of slides, but there is a spectrum. Now, this repeat size, there's an intermediate state between somewhere of repeat sizes of in your 30s up to 54, that you will have some onset of symptoms like maybe restless leg. Now, if you have a 55 repeat or bigger, generally speaking, you will have onset of ataxia, SCA3, ataxia motor impairments. The bigger the repeat, the earlier the onset. Doesn't necessarily mean the more severe your disease, just means often earlier the onset. The repeat size, and there's some really interesting science that's coming out that it's not just the size that you inherit from your mom and dad, it's the size that expands throughout your lifetime. We call this as somatic instability. It's a fancy word for basically that repeat getting longer in regions that are vulnerable. And so you would see this in the Huntington's field and a lot of these other repeat expansion fields. And so that remains to be well established in SCA3, but I would put my money on it being a pathogenic mechanism of disease.
So we know that repeat expansion is inherited, but does that mean everyone in the family is going to have the same repeat length?
No. So you inherit from either your mom or your dad one copy, right? Each parent provides one copy of this gene, generally speaking. And if mom had the SCA3 disease gene and you happen to get that copy from them, it might be a little bit smaller, a little bit bigger, because again, mom's eggs has been sitting inside of her for years and years, and modifying and changing as they developed. And so just like even in our model systems, we have to track our mice and how those repeat sizes modify through the generations. The same thing as patients. It's not going to hyper expand dramatically, but it will have anticipation in many cases with an expansion of the repeat over time.
And so I know from my experience that sorry, can you explain a little bit how what anticipation is and how that's related to repeat length?
Yeah, so anticipation is just the fancy word for basically when you gain a little bit bigger repeat size over the generations. So as mom passes the gene or dad passes the gene, you might get a little bit bigger repeat size.
This is kind of an interesting question that asks, is the onset of symptoms related to how much of that protein has built up in the brain? And with an ASO therapy, how does that help actually clear the protein from the brain that's already built up over lifetime?
That's an awesome question. I wish you were in my lab. It is not directly related to the accumulation per se. And that's evidenced again through the model systems. Now, nuclear accumulation or aggregation of this protein is a hallmark of this disease. You will see it in regions that are affected. Whether clearance of that aggregated protein is sufficient for therapeutic rescue of phenotypes is not actually well established to date. In some evidence, it is in model systems, but it has not been tested in the pipeline towards preclinical development or phase 1 clinical trials yet. The accumulation of proteins, though, even with the ASO technology or any sort of gene therapy approach, is that you're alleviating the burden. So by cleaving this protein, the general garbage dumps of a cell, like something known as autophagy or the proteasome, will clean up this mess. And so that is allowing for what's known as kind of a holiday or a hiatus in the cell, relief from that burden of this aggregated protein that's blocking up the rest of the garbage dumps from flowing out.
All right. Well, I wish we had more time to go through more questions, but we are at the end of the hour. So once again, I wanted to thank Dr. McLoughlin for joining us today and providing us with so much information. This really is an exciting time for SCA3 and drug development.
I thank you for the opportunity and all of your attention. I wish I had more time to answer questions, but see you all at an Ataxia conference in the near future.
And Hayley will be joining us at the AAC meeting in the end of March. So hope to see you all there.
Yeah, come say hi, please.